of insulin activity in the central nervous system. So how this talk is going to look, I'm going to give a little bit of background information, give an introduction to Alzheimer's disease, tell you about some of the risk factors and the relationship between type 2 diabetes and Alzheimer's. I'll mention my hypothesis and the methodology I use to obtain the select results I'm going to be showing today, some conclusions, implications, and then I would like to thank a couple of people. Okay. So Alzheimer's disease is the most common neurodegenerative disease worldwide, and it currently affects more than 44 million people. It's characterized by the loss of memory and decline in cognitive function in people who suffer from Alzheimer's disease until they eventually return to an infant-like state, relying solely on other people to help them with the everyday tasks that they could once perform on their own. At the cellular level, Alzheimer's disease is characterized by extracellular amyloid beta plaques, or AB plaques, and necrotic fragments of disconnected neurons called neurofibrillary tangles, or NFTs. The appearance of these cellular hallmarks leads to chronic activation of the glial cells, mainly microglia, which are needed to maintain and return the brain to homeostasis during infection and in disease states. But in Alzheimer's disease, the microglia cannot destroy the plaques and tangles, and therefore their activation persists, leading to chronic activation of microglia, which is detrimental to surrounding neurons and glial cells. There are a number of risk factors for Alzheimer's disease, some of which are non-modifiable, like age and genetics, and some which are modifiable, such as obesity and smoking. Among the risk factors for Alzheimer's disease, one of the recently emerging risk factors is type 2 diabetes. So I think a lot of you are probably familiar with what type 2 diabetes is, but I'll just briefly mention that it's characterized by high glucose concentrations in the I'm not touching the buttons. In the blood due to functional insulin resistance. And people with type 2 diabetes also experience other complications like chronic inflammation, dysregulated lipid metabolism, and a number of other things. So type 2 diabetes currently affects more than 350 million people globally, and the prevalence of type 2 diabetes is on the rise. And since type 2 diabetes is a risk factor for Alzheimer's, this is going to inevitably lead to an increase in the incidence of Alzheimer's as well. Insulin has also been shown to be very important in the central nervous system, in addition to its role in the periphery. It's important in the brain for learning, memory, and cell survival, and it is shown to be neuroprotective in the CNS. In fact, there's currently clinical trials going on right now, such as the SNF clinical trial, where insulin is used in an intranasal administration, and it's been shown to alleviate symptoms of mild cognitive impairment, which is basically pre-Alzheimer's disease often. So one mechanism, and don't worry about this, there's not going to be a quiz later, one mechanism by which insulin is protective in the brain and can inhibit Alzheimer's disease is through the PI3K pathway, which physically blocks some of the hallmarks of Alzheimer's disease that I mentioned. So I just want to stop here and mention that this is, of course, not the complete pathway. This is a very simplified version, and the actual PI3K pathway in the body is much more complex, with arrows leading in and out in addition to what I have shown here. But as you can see here, when insulin binds to its receptor, it activates the PI3K pathway, and it inserts these blocks which otherwise would lead to the formation of these plaques and tangles. So because these blocks are in place because of the insulin, the plaques and tangles, which are hallmarks of Alzheimer's disease, are put into place, and it has potential to eliminate the hallmarks and therefore eliminate Alzheimer's disease. But in type 2 diabetes and in people with Alzheimer's disease, the insulin signaling is not fully functional, and so you have less binding of insulin to the receptor, and when you do have insulin binding to its receptor, you have less activation, thereby removing these blocks, and you have the pathological formations in Alzheimer's disease showing up. So my research has been looking at the relationship between type 2 diabetes and Alzheimer's disease, and myself and previous researchers have identified that reduced insulin signaling as well as increased inflammation, which are common in both type 2 diabetes and Alzheimer's disease, may be the mechanistic link between type 2 diabetes and Alzheimer's. Where I specifically am inserting my research is in this knowledge gap between insulin signaling and inflammation. So is there a relationship between the two? And this in the central nervous system has really not been studied very much. And so I am specifically looking at the effect of insulin on the immune cells of the brain, the microglia. Which brings me to my hypothesis. So I've hypothesized that the neuroprotective properties of insulin may be in part due to, first off, the modification of cytokine secretory profile from glial cells, and also due to a reduction in the glia-mediated toxicity towards neurons. So I'm going to go over the methodology. The first one's pretty simple, and you've probably all used this in your time. But the first step in testing this theory was to check that the insulin receptors and insulin signaling molecules are present in glial cells of the CNS. So of course I extracted mRNA from my cells of interest, converted it to cDNA, and then I both designed and purchased primers for my gene products of interest, and then ran PCR and imaged them on with gel electrophoresis. And this is the result I got. And this is why I wanted the lights dimmed. I don't know if you can see all the bands there. If you're having a difficult time seeing, because I know sometimes it is difficult on a screen, some of this is recently being researched, so I can give you a link to my paper if you're interested. But so the 4 cell types that I'm showing here that I was interested in looking at are THP1 monocytic cells, which are often used as a microglia model, U373 astrocytic cell line, which we commonly used in our lab as an astrocyte model, primary human astrocytes, and also primary human microglia. I also did this on the SHSY5Y cells, which we use in our lab as a neuronal model, and I confirmed the presence of these signaling molecules. So you can see, hopefully, where is this? You can see along the top that there's 3 insulin receptors, the general insulin receptor, and then it comes in 2 isoforms, A and B. You can see insulin receptor substrate 1 and 2, which are the primary messengers on the inside of the cell. The insulin-like growth factor 1 receptor, which insulin also signals through, and the G3-PDH, which is our housekeeping gene. So hopefully you can see all the bands there, but in the 4 cell types, I found that all the signaling molecules to be present in these cells. So once I had confirmed that the necessary machinery was there for insulin signaling in glial cells, I moved to our system, which models neuroinflammation. So our system is a one-way modeling system to look at glial to neuronal signaling in the brain and to kind of tease out individual effects rather than a whole in vivo model, which it can be difficult to pinpoint where different things are coming from. So this is similar to the model that Stephanie used. So on day 1, I plated the microglia, and after a period of 15 minutes rest and recovery, I would add the cytokine cocktail to induce an inflammatory-like state and the insulin. I let the microglia incubate for 48 hours, and on day 2, I plated the neurons and allowed them 24 hours to adhere to the plate. The following day, I would collect supernatants from the microglia and run an ELISA, so enzyme-linked immunosorbent assay, to test for different cytokines, and at the same time, I transferred cultured media or supernatants from the microglia onto the neuronal cells. After the cultured media was transferred onto the neuronal cells, I waited 72 hours and then I did an MTT assay to test for the cell viability of the neurons. And these are the results that I obtained. So we found